Hello, tonight we're going to talk about photosynthesis. The organelle that photosynthesis occurs in is the chloroplast. So let's review a little bit about the structure of a chloroplast. Do you remember the chloroplast had two membranes because of the theory of what? Endosymbiosis. On the inside are stacks of little round discs called thylakoids. A stack is called a grana. And then there's fluid in between them, and the fluid is called stroma. Different parts of photosynthesis will occur at different regions inside the chloroplast. So as we go through, I'll remind you of those things. The formula for photosynthesis, and yes, you do have to know this, is 6H2O plus 6 carbon dioxide yields 6 oxygen and glucose, which is C6H12O6. Inside the chloroplast, you have several different pigments. There are chlorophylls. There are actually 5 different types of chlorophyll. Chlorophyll A, B, C, D, and the one I can't remember. It's not E. And then the carotenes, which are orange and yellows, anthocyanins, which are reds and purples. There are 2 main types of chlorophyll, chlorophyll A and chlorophyll B. Chlorophyll A is kind of the most important one out of all of the molecules in there. All of these different pigments are found in piles. A pile of pigments is called a photosystem. And at the very top of the photosystem, you'll have a chlorophyll A molecule. All of those different pigments absorb different frequencies of light. Like chlorophyll A absorbs purples. It reflects greens. Chlorophyll B absorbs blues and purples, and it reflects green. Carotenoids absorb a lot of blues, and they reflect yellows. The anthocyanins are going to be in here someplace, and they reflect the reds and the purples. So they all have different absorption spectrums and reflection spectrums. In bacteria, because they do not have they don't have a photosystem. Actually, they have photosystem. They don't have the chloroplast. They don't have any organelles. There are still some bacteria that go through photosynthesis. And those bacteria do have a membrane on the inside of their cell that is similar to a thylakoid. It's very simplified. On that membrane, they have a photosystem. It's actually photosystem 1. They go through a process of cyclic photophosphorylation, which is a very simplified photosynthesis. What happens is sunlight comes down and it hits a chlorophyll molecule. That chlorophyll molecule gets real excited, and when it gets excited, the electrons absorb the energy, and that causes an electron to jump to an outer electron shell. When it does, it either gets stolen by an electron acceptor, but in this case it's not going to be. It'll fall back to its normal position. When it does, it gives off light, or it gives off heat, or something like that. Well, when it does give off that energy is converted and stored as an ATP molecule. So bacteria create ATP by using sunlight coming down, hitting a chlorophyll molecule, the electron jumping to an outer shell. When it falls back down, that energy can be converted to ATP. So that's cyclic because nothing is lost from the molecule. The electron stays with the same molecule. It just runs around in a little circle. Okay, that pile of pigments, like I said, is a photosystem. There are 2 photosystems, photosystem 1 and photosystem 2. Photosystem 1 was discovered in bacteria, and so it was named photosystem 1. And then in plants, they found photosystem 2 and photosystem 1, both of them occurring. However, they don't occur in that order. Photosystem 2 occurs before photosystem 1 in the process. It's not a big deal. It was just named second. So don't let the 1s and 2s freak you out because they aren't in order. It's okay. Okay, so what's going to happen is a ray of light, a photon of light, is going to come down and hit the photosystem. One of those pigments will absorb that frequency of light. Not all of them absorb the same frequencies of light, and because they don't, somebody in the pile does. Well, somebody absorbs it, and then it transfers the energy to another molecule, and then to another pigment, and then to another pigment molecule. Eventually, that energy ends up at chlorophyll A. When it does, then we're going to start the rest of the process. But it kind of jumps around on this photosystem until it hits chlorophyll A, and then things really start. So here it is. The photon comes down, hits one molecule, bounces around until it finds chlorophyll A. Okay? So, in photosystem 2, the light comes down and it bounces around, and the chlorophyll A that it hits is a very specific chlorophyll A. It's pigment 680, which means, this P680, means it absorbs the frequency of light of 680 nanometers. That's just what it absorbs. It's a specific frequency. So, the energy hits chlorophyll A. Chlorophyll A gets so excited the electron jumps to an outer shell. Well, when it jumps to an outer shell, another molecule called an electron acceptor grabs the electron. It steals them. While that process is going on, another photon of light comes down and it hits a water molecule. Well, when it hits the water molecule, that water molecule is going to split. The oxygen is going to be given off, and that's what plants, where the oxygen comes from that plants give off, that you breathe in. The electron is going to replace the electrons that the chlorophyll A pigment 680 lost, and the hydrogen we'll see in a little while. So the water splits, oxygen leaves, hydrogens are going to hang out, the electron replaces the other electrons. Now, whatever happened to that electron that was lost? Well, it's going to go down through an electron transport chain. An electron transport chain is a series of enzymes that are one right after another in the thylakoid membrane. Okay, so remember the thylakoid, the portion of the chloroplast? Okay, it's going to go through a process of chemoosmosis in the production of ATP, which is called photophosphorylation. Photo means light. Phosphorylation means to attach a phosphate group. So in the process of using light, we're going to create ATP by attaching a phosphate group to ADP, adenosine diphosphate. So let's look specifically at this electron transport chain. As the electrons move from one molecule to the next molecule to the next molecule, hydrogen, that hydrogen right there, is going to be pumped across the thylakoid membrane to the interior of the thylakoid membrane. When it's pumped across against the concentration gradient, that electron will be moved down from enzyme to enzyme. At the very end, and it's not pictured here, but at the very end is a specific enzyme called ATP synthase. When those hydrogens build up on the inside of the thylakoid, it's kind of like water behind a dam. When they're back there, they build up a lot of pressure. Well, those hydrogens move back through ATP synthase. As they move back through to find equilibrium, ATP synthase creates ATP. So we are now at that point where ATP is going to be produced. So hydrogens are pumped across, come back through ATP synthase, create ATP. Now that electron hits the next photosystem. At the base of that picture would have been photosystem 1. That electron is going to bounce around photosystem 1 until it hits chlorophyll A, a different chlorophyll A. This one is pigment 700. The first one was pigment 680, so this is a slightly different molecule. Another photon of sunlight hits this photosystem. It's going to bounce around and hit this same chlorophyll A. It's going to cause that chlorophyll A pigment 700 to lose its electron. The electron that it lost is going to be replaced by the one that just came down through the electron transport chain. Now the electron that pigment 700 lost is going to go to another primary electron acceptor. So it was stolen again. Okay, so sunlight hit, bounced around, pigment 700 lost its electron, this electron replaces it, Everybody has the right number of electrons. But we've got this electron acceptor hanging out with some extra electrons. Well, at this point, the electron acceptor is going to give its electron plus a free hydrogen from that water that was split to a carrier molecule called NADP. Well, those two things, the electron and the hydrogen, attach and they form an NADPH molecule. This molecule is going to be used during the Calvin-Benson cycle, which is the second half of photosynthesis. This is the first half. Okay, we're going to watch a short little video right now, so pause it. Okay, water floating in the air is going to come, carbon dioxide comes in through the stomata, comes to the chloroplast. We're going to watch the reactions occur inside here. Those are all photosystems. Okay, photons of light are going to hit the first photosystem. Electrons are going to be carried over to the electron transport chain. Electrons then are going to be carried over to photosystem 2. Photosystem 2 is going to bind it here with an NADP carrier molecule. Here's the water being split, hydrogen. Here's the ATP synthase molecule right here, creating ATP, which is all the little stars. Now here comes the Calvin cycle, which we haven't gotten to. Actually, we'll stop it here. Okay, so this is the overall process. You can see the light reactions. Light comes down, bounces around photosystem 2, water split, the electron is stolen, electron replaces the one. Comes down the electron transport chain, we're going to end up with ATP synthase, creating ATP. Another photon hits photosystem 1, bounces around, electrons are stolen, electrons are replaced. Here's a short electron transport chain, bringing in NADP with a couple of hydrogens and 2 electrons, creating NADPH. All of this is occurring in the thylakoid, and as you saw in the short little video, each photosystem is on top of a protein on the thylakoid, and then those molecules are going to be carried right along in the interior of the thylakoid membrane from photosystem 1 to the cytochrome complex, which is the electron transport chain, to from photosystem 2 to photosystem 1, and then you have your NADP reductase, which is the enzyme creating your NADPH molecule. And don't forget your ATP synthase at the very bottom. All those hydrogens moving back through, making lots of ATP. So now we're going on to the Calvin-Benson cycle. These are called the light-independent cycle phases because these don't have to have sunlight to occur. They occur 24/7, but it does not require sunlight. Unlike the first one, which required sunlight, everything started with those photons of light from the sun. Okay, this is a little bit more complex. we're going to do 3 at a time. 3 carbon dioxides come in, bind to a molecule called RUBP, ribulose bisphosphate. When those bind, they create 6 3-phosphoglycerates, which are 3 PGAs. In comes some of that ATP that we made in the light reactions. Okay. Then NADPH is going to come in and drop off its hydrogens, 6 of them, drop off 6 hydrogens. The ATPs dropped off phosphates. See, the phosphates have been added to your molecule. Then you drop off some hydrogens, and those reorganize into 6 glyceraldehyde 3-phosphates, which are 3G3Ps. I can't even say it. Okay, one of those breaks off. The other 5 then reorganize again and become ribulose bisphosphate again. Then that's going to come up, grab some more carbon dioxide, do it again, grab some phosphates from ATP, grab some hydrogens from NADPH, reorganize, and drop off a second G3P. Now 2 of these G3Ps are going to recombine to create a glucose molecule. And that's the end. So we have to do this twice, and then we're done. Okay, so that's the whole process. Now some plants, while doing the Calvin-Benson cycle, do something prior to that to help them out, because they live in environments where conditions aren't exactly perfect. So they have to have some alternative methods to help them. For example, in the desert, if a plant has their stomata open so they can get carbon dioxide, they're also losing water through transpiration. Desert plants can't afford to lose any water. And so they have to do something to bring the carbon dioxide in while having their stomata closed most of the time. So we're going to find out about those things. So there are 2 special processes, C4 and CAM, and we're going to talk about both of these. C4 plants are things like grasses and corn, and they have a different leaf anatomy. Instead of the normal leaf anatomy, they have cells called bundle sheath cells, and then they have mesophyll cells. And they're kind of, let's see if I have a picture. Yeah, I do kind of. Okay, they have a vein right here. Then they have their bundle sheath cells, and then they have their mesophyll cells. And then out here are air spaces for carbon dioxide, oxygen, and water exchange. But they're still inside the plant leaf. Okay, so it's a little bit different. So their anatomy is a little different. What they need to do is they bring in carbon dioxide through the stomata. Then they're going to have the carbon dioxide enter a mesophyll cell, and immediately it's converted to a molecule called malate, which is a 4-carbon molecule. That's where that C4 comes from. It's a 4-carbon molecule. That process is called fixing carbon dioxide, fixing it in a different type of molecule so that it's no longer present in the cell. When the cell then opens its stomata again, there's no carbon dioxide inside the plant, inside the leaf. So carbon dioxide diffuses in. It fixes that carbon dioxide, and then the stomata can open and more carbon dioxide come in. Other plants don't do that. They only bring in carbon dioxide that they need. As a result, C4 plants bring in more carbon dioxide and make more sugar than other plants do. So they're actually more efficient. They're more advanced plants. Okay, once that malate is in the mesophyll cell, it's then moved into the bundle sheath cell. When it gets to the bundle sheath cell, it's converted back into carbon dioxide, but immediately goes into the Calvin cycle, creating glucose. So it does this in 2 different cells, which is different from normal photosynthesis, where it just occurs in 1 cell. And it's extremely productive in places where it's very hot, but not a desert. So if you think about it, where do we grow corn? Well, we grow corn in the Midwest. What's it like in the summertime? 90, 100 degrees? Well, it's kind of hot. Okay, so their unique thing is they have 2 cells, and then the sugar immediately goes into the vein. Okay, so we're going to take a look at that one.
In the leaf tissue of C4 plants, the Calvin cycle occurs in one type of cell, while carbon dioxide is captured in another type of cell. The mesophyll cells are closest to the air spaces and contain the enzyme PEP carboxylase. Unlike rubisco, PEP carboxylase does not bind to competing oxygen molecules, so this enzyme is always available to capture carbon dioxide, even when carbon dioxide levels are very low. PEP carboxylase fixes carbon dioxide in mesophyll cells. The 4-carbon organic acids that result travel to the bundle sheath cells, where rubisco is located. The 4-carbon organic acids then release carbon dioxide into the bundle sheath cells. With relatively high levels of carbon dioxide available, the rubisco in these cells catalyzes the Calvin cycle with carbon dioxide, rather than catalyzing photorespiration with oxygen. In this way, the C4 plant can produce sugars efficiently even in hot, dry weather, when stomata are closed and carbon dioxide levels are relatively low throughout the plant. Soybeans, rice, and potato plants are examples of C3 plants, which have the Calvin cycle but lack the C4 cycle. Corn, crabgrass, and sugarcane are examples of C4 plants, which have the C4 cycle in mesophyll cells. The resulting 4-carbon molecules travel to the bundle sheath cells and liberate carbon dioxide, which initiates the Calvin cycle. Another type of carbon fixation strategy has evolved in cacti and a variety of other succulents. This mechanism is called CAM. CAM is an abbreviation for crassylation acid metabolism. In this pathway, a variation of the C4 cycle captures carbon dioxide at night, while the stomata are open. The 4-carbon molecules produced are stockpiled, so that in the heat of the day, when the stomata are closed, the Calvin cycle can receive carbon dioxide from the stockpiled molecules. In this case, the C4 and Calvin cycles are separated in time, but the cycles are both present in the same cells.
So let's go to the CAM, the crushylene acid metabolism process. Now, they just described that a little bit. It's found in desert plants. The problem within the desert is that during the daytime, it is so hot, if the stomata are open, the water will transpire out of the plant and the plant will dehydrate and die. So they only open their stomata at night when the temperatures are cooler and water will not transpire quite so rapidly. So they have had to create a process that allows them to still have carbon dioxide during the daytime, but not have their stomata open. And so that's what they do. During the nighttime, the stomata are open, the carbon dioxide is pulled in, converted to crushylene acid, and then during the daytime, the stomata are closed, crushylene acid is converted back to carbon dioxide, and it enters the Calvin cycle. So it's very similar to C4 in that they fixed carbon dioxide, where C4 does it in a different type of cell, bundle sheaths and mesophyll cells, as opposed to the CAM plant where it occurs in the same cell, but at different times of the day. One's at night, one's during the daytime. Okay, so carbon dioxide comes in, turn to that crushylene acid during the daytime, turn back to carbon dioxide, and then go through the Calvin cycle making G3P. So this slide puts both of them together so you can see the difference. Mesophyll cells, bundle sheath cells, so this is C4. Here's the CAM. It's that organic acid, crushylene acid, converted from night during the daytime, go through the Calvin cycle. So it's a temporal separation, which is timing, as opposed to a spatial separation, which is space, different cells. Okay, and these two slides, this one and the next one, kind of help you to see the difference between them. 2 cells versus 1 cell, fixes it to malate versus crushylene acid, keeps the carbon dioxide levels high and increases sugar production. They do it because they have to keep the stomata closed during the night, different types of plants. And This is a C3 plant anatomy. It's got the various cells. These are called palisade mesophyll cells. These are spongy mesophyll cells, both of which have lots of chloroplast in them. This is where photosynthesis is occurring. If you look at the C4 anatomy, very different type of anatomy. You've got your mesophyll cells here, your bundle sheath cells here, and here's your vein. So it's kind of more of a radial type pattern. Okay, that's it for tonight. Have a great night.